Hello, everyone. Welcome to lecture Carbohydrate Metabolism 1 in our comprehensive Carbohydrate Metabolism series. Having thoroughly mastered in chemistry of carbohydrates, their structures, isomerism, ring forms, anomers, meta-rotation, and the complex polysaccharides, including glycogen, cellulose, and glycosamine glycans, we now transition seamlessly into their metabolic fate in the human body. Today, we focus on one of the most clinically relevant topic in biochemistry, the digestion and absorption of the carbohydrates. This lecture is foundational because disorders at any step, from enzyme deficiencies to transport defects, manifest as common clinical conditions such as lactose intolerance, malabsorption syndromes, and even contribute to metabolic syndromes. Understanding these processes at the molecular level equip you to diagnose, manage, and explain these disorders to patients. So let's proceed systematically with the emphasis on both biochemical mechanisms and their clinical correlations. So let's define carbohydrates and their nutritional role, right? Carbohydrates are literally the hydrates of carbon with the general empirical formula that is CH2ON, right? Their primary nutritional role is to serve as a major source of energy in the human body. Digestible carbohydrates yield approximately 4 kilocalories per gram upon complete oxidation. In clinical practice, this is why carbohydrates continuing on counting is the central to diabetic management and why total parenteral nutrition formulation are carefully balanced with glucose equivalents. Undigested carbohydrates, however, reach the colon and can cause osmotic diarrhea and be fermented by gut microbiota. A key point in irritable bowel syndrome and small intestinal bacterial outgrowth or overgrowth, right? That is SIBO, right? So now let's move to the photosynthesis, the origin of dietary carbohydrates. All dietary carbohydrates ultimately trace back to photosynthesis, right? As you can see on your screens, the basic formula to get the basic of the basic carbohydrate structure or the formulation, right? CO2 plus H2O in the presence of energy or the sunlight that yields C6H12O6 and the release of oxygen, right? So this slide reminds us the sun energy captured in the plant glucose because the fuel for human metabolism. In medicine, this underscores why plant-based diets rich in complex carbohydrates are metabolically advantageous. They deliver energy slowly and are accompanied by fiber that modulates absorption and gut health. So let's move to the classification of the carbohydrates or the dietary carbohydrates. So they are classified as the monosaccharides, the disaccharides, and oligosaccharides, along with the more complex form. Those are polysaccharides. Right? Starch and glycogen are the predominant forms in our diet. This classification is clinically important because they are more tend to have the deficiencies, right? Along with the deficiency produce highly specific symptoms, right? The starch, the major dietary polysaccharide. As you can see on your screen, the complex structure of the starch, right? The starch is the chief storage carbohydrate in the higher plants and supplies approximately 80% of the dietary calories worldwide. It consists of two components, the amylose and the amylopectin. Amylose, the linear chain of glucose linked by alpha-1,4 glycosidic bonds, and the amylopectin are the branch polymer with the alpha-1,4 chains and alpha-1,6 at branch point at every 24 to 28 residues, right? So clinically, branch structures of amylopectin allow faster enzymatic attack, explaining why refined starches raise blood glucose levels more rapidly than the unprocessed plant starches. In patients with the diabetes or reactive hypoglycemia, choosing starches with higher amylose content helps blunt postprandial glucose spikes. Now, the glycogen, that is the major storage form in animals, right? The liver and the muscles are the main sites of its storage. It is structurally similar to the amylopectin, but more highly branched, as alpha 1,6 branches occur every 4 to 8 glucose residues. This extensive branching creates multiple non-reducing ends, permitting rapid mobilization of glucose during exercise or fasting condition. So, what is the clinical correlation at this very point by having glycogen? So, defects in glycogen breakdown, that is glycogen storage disorders or diseases, or excessive accumulation leads to hepatomegaly, hypoglycemia, and muscle cramps. So, as you can see on your screen, the diagram on this slide beautifully illustrates the alpha 1,4 backbone and the alpha 1,6 branch points. So, memorize this structure as it appears in every single metabolic pathway diagram, right? Now, let's move to the overview of the digestion of the carbohydrates. Digestion of carbohydrates is a sequential enzyme catalyzed hydrolysis of glycosidic bonds, beginning in the mouth and completing in the small intestine. No significant carbohydrate digestion occurs in the stomach. The end products are monosaccharides, the glucose, fructose, or the galactose that can be absorbed. So the process of digestion begins at the level of mouth, right? The salivary amylase, the ptyalin, initiates starch digestion. The optimum pH, that is 6.7, the action of amylase that that hydrolyze the internal alpha 1,4 glycosidic linkages, and the products of this enzymic activity are the maltose, maltotriose, and alpha limit dextrins. Its action is brief because it is inactivated at the acidic pH of the stomach that is less than 3. So, clinical note at this very point is that the patients with the xerostomia, the dry mouth, and salivary gland dysfunction, oral carbohydrate digestion is impaired, though pancreatic amylase compensate this condition adequately. So, next is the mouth. No carbohydrate splitting enzymes are present in the stomach. The acidic environment, the pH of it is approximately 1 to 3, completely stops salivary amylase activity. This slide emphasizes the gastric secretion is irrelevant for carbohydrates, a common point tested in exams, right? The next step is the duodenum. Pancreatic amylase, the amylopsin, that is another name of pancreatic amylase, is secreted into the duodenum. The conditions required for its activity are the optimum pH, that is approximately the 7.1. The bicarbonate-rich pancreatic juice neutralizes chyme. That is a semi-solid food, right? So the action continues hydrolysis of alpha 1,4 linkages, producing maltose, maltotriose, and limit dextrins. So the clinical importance of this pretty point is this, that the pancreatic exocrine insufficiency in case of chronic pancreatitis, cystic fibrosis, pancreatic resection, markedly reduce amylase activity, leading to steatorrhea and carbohydrate malabsorption. Stool tests for fecal elastase or serum amylase or lipase help to diagnose this condition, right? So next is the small intestine. Brush border enzymes. Final digestion occur at the brush border of the enterocytes in the jejunum and the ileum. The intestinal amylase that changes the maltase into 2 glucose molecules. The sucrose converted to glucose and fructose in the presence of sucrase. The lactose molecule get converted into glucose and galactose in the presence of lactase, right? Isomaltase. that convert the isomaltose and alpha-dextrins to glucose molecules. These are membrane-bounded enzymes. Any deficiency here produces osmotic diarrhea because undigested disaccharides draw water into the lumen and are fermented by colonic bacteria, producing gas and short-chain fatty acids. Next is the absorption of the simple carbohydrates. Those are get converted by the enzymatic activity present in the gut. All carbohydrates are ultimately broken down to monosaccharides, glucose, fructose, galactose, and some of the simple other carbohydrate structures or the monosaccharides. And they are completely absorbed in the small intestine, predominantly in the jejunum and duodenum. Right? The rate of absorption decreases from proximal to distal small intestine and depends on the specific monosaccharide. For example, glucose is more rapidly absorbed, more than galactose, and that get absorbed more frequently than the fructose molecule. Right? So what is the mechanism of absorption behind the absorption of the simple carbohydrates? Absorption involves three mechanisms: the simple diffusion, facilitated transport, and active transport. Now let's discuss a small simple diffusion, the minor for the fructose at higher concentration. For example, the facilitated transport, the second mechanism, that is carrier mediated and energy independent mechanism. And the third one is the active transport, that is energy dependent and sodium coupled. Receptors are involved in the absorption of the simple carbohydrates or the monosaccharides. So key transporters here are the sodium glucose link transporter 1, SGLT1, that is active transporter for glucose and galactose, that is sodium dependent. And the second transporter, the key transporter that is involved here is the GLUT2, that is facilitated diffusion of the glucose, galactose, and fructose across the basolateral membrane into the blood. The next one is the GLUT5, GLUT5, right? So fructose specific facilitated transporter on the apical membrane. So what is the clinical correlation at this very point regarding the key transporters involved in the absorption of the simple monosaccharides units or simple carbohydrates? Congenital glucose-galactose malabsorption, that is SGLT1 mutation, presents with the severe watery diarrhea from birth. In diabetes, GLUT2 and SGLT1 expression is upregulated, contributing to hyperglycemia. Now, the enzyme deficiencies and clinical disorders. This is the most clinically significant slide at this very point regarding this lecture series or this very session, right? So very first one is the lactase deficiency or the lactose intolerance. Most common worldwide, especially in Asian and African populations. Undigested lactose cause bloating, flatulence, abdominal cramps, and osmotic diarrhea. So management for this condition is lactose-free diet, lactose supplements, or yogurt that contains bacterial beta glucosidase, right? The next enzymic deficiency is the sucrase isomaltase deficiency. That is very rare. Presents with diarrhea after sucrose ingestion, right? The third one is the disacchariduria or disaccharide deficiency. In this condition, urinary excretion of the disaccharides due to severe mucosal damage occurs, right? So that is the pretty condition that one can experience in a celiac disease, right? The fourth one, the last one, is a monosaccharide malabsorption. Glucose-Galactose Malabsorption syndrome, that is life-threatening condition in the neonates. So these disorders, these disorders highlights why a detailed dietary history is essential in patients presenting with the chronic diarrhea and failure to thrive in neonates and the individuals those are having the enzymic deficiency. So lactose intolerance, sucrose deficiency, disaccharide deficiency, and monosaccharide malabsorption syndrome are very lethal sometimes if they are untreated. So we have now completed a thorough examination of the carbohydrate digestion and absorption, from salivary amylase to brush water enzymes and specific membrane-bounded transporters. You now understand why certain sugars cause symptoms in specific enzyme deficiencies and how these processes integrate with the overall energy metabolism. In the next lecture, that would be carbohydrate metabolism 2, we will enter the cell and explore glycolysis, the central pathway for the carbohydrate metabolism. So thank you very much for watching. In this lecture, if this lecture clarified the biochemical and clinical aspects of the carbohydrate digestion and absorption, please like the video, share it with your fellow medical students and colleagues, and subscribe to the channel for the complete high-yield medical biochemistry series. Turn on the notification bell so you never miss the upcoming lectures on the glycolysis or the glycogen metabolism and beyond, right? So drop your question if you have any or clinical cases in the comments. I personally read and reply to them. Which topic would you like me to cover in the greater depth? So this is Dr. MBBS signing off. Stay focused, keep thriving, and keep revising the enzyme deficiencies and transporter daily, and keep mastering biochemistry with precision and passion. See you in the next lecture. That would be on carbohydrate metabolism 2. Thank you once again.